that leaves us one full week in week 10, which will include some review time on Wednesday, December 3rd. So, and we can only have one class the following week, which is the final exam on the 8th, which means we're right down to it. Half a week next week and half a week at the end, and one more full week after today. So, on the one hand, that means you're almost done. On the other hand, that means to get there, keep in the zone of completing the assignments, studying for the quiz, asking questions, and staying with the work that you've done this far. And likewise, check the Moodle grades and make sure that you got one in there for everything that you handed in. Or check to see which thing I don't have. And there's an opportunity to complete that, up until the exam. And if you need more time, talk to me. Some concern about that, right? We can be very flexible. The goal is that doing the work makes the learning. Being here, doing the work, answering the questions. And so that's what we're trying to get. So today, I just passed back everything that I have to date. So you've got it all to study from now. And I want to start there. We spent some good time on Monday reviewing photosynthesis and respiration, and we can continue based on now the things that you have in your hand, if there's still more questions, or I can get right into the hormones, tropisms, and plant responses. So I'll leave it open for your questions, and then we'll move on whenever we get through that. I'll get that. Let me make a couple specific comments in that case. So keep in mind here, Calvin cycle is photosynthesis, citric acid or Krebs cycle is respiration, different processes. But the components are similar because it's the two directions of the equation. So the same factors are at play, just trying to keep track of where they are and what they do. Here, I wanted to especially call your attention in respiration to the importance of, over here was the electron transport chain, but mainly that oxygen, right, role here, which is also a big limiting factor, which if you don't have it, then you get lactic acid fermentation or alcohol fermentation, anaerobic respiration, and significantly less energy output. So keep that one in mind, right, as both the fuel for the fire of respiration, the energy harvest fuel, and as doesn't appear until the third step. Right? So all this can still happen, and you get that couple ATP, small energy output, that comes from anaerobic to until you get here, and then there's more opportunity for payoff, or the process ends if oxygen's not there. So that's the big player there in respiration. That oxygen provides energy, and the oxygen comes from the atmosphere, from the air. So I just talked about this one. Aerobic is definitely higher energy, right? More efficient. And then down here in the graphs, show the relationship between respiration rate and temperature. Just keep in mind, I'm talking specifically about things that I've shown you photosynthesis exceeds respiration rate and the plant grows, right? This is all potential for growth then. When respiration rate exceeds photosynthesis, now we're consuming our available stores without replenishing. Right? Now the total quantity of food in the fridge is going down, and we're not growing. Here, we're stocking the fridge and simultaneously adding growth. So just try to make sure that that much makes sense. No others? All right. So then I'm going to start in on the next one. Again, the quiz will just be photosynthesis and respiration on Monday. What do you guys want? I've got an assignment that goes with today's lecture that I was planning on assigning on Monday for the following Monday, but I can give it today and still make it for that too. But I was just trying to keep like, okay, focus on the quiz and then move on, but open to your preference.
Would it still be due, like if it was given today, would it still be due?
And so we do Monday, December 1st.
Works for me.
Sound good? In that case, pick it up on your way out. Nice. And we'll add reading, and believe it's doing that off the top of my head, so double check that. Yes. So here, I would suggest, in the interest of keeping the quiz material straight, unless you feel comfortable, confident there, that's excellent. But listen today, take it in, take notes, and then I would suggest after the quiz next Monday, do the reading for it, review again, and complete the homework. Try to keep it compartmentalized and moving along. Let's begin. Plant hormones. So we're building up more complexity, organ systems. Now we've included how the plant works, right? Both how it makes energy and how it uses energy. These important processes, the effects related to stomata, to environmental condition, right, like humidity, direct light, water supply. And now we're talking about internal complexity of how the plant is signaling a bunch of these processes that are powered by the food from photosynthesis as it's broken apart to get the energy in respiration. So in a very general sense, plant hormones, like mammalian hormones, are chemicals that regulate growth processes, in this case, specific plants. They're effective in small quantities, so doesn't have to make a lot of them to get the effect. They're mostly found in the root tips, the shoot tube tips, and at nodes, which sounds familiar. Natasha? Meristematic tissue. That's where these things are being made, at those same areas. Because as the meristem develops into whichever one of the tissue types it is, then it's right at that site where it can be influenced by these things, or if the plant needs to make more hormone, it can also do that. And this is one generally studying plant hormones it's still open. We don't know everything. We don't know all the plant hormones or how they work exactly. Not every pathway is mapped yet or anything. So it's still like an exciting research area. And so here I'm just indicating meristems is where we find these things. And it's useful to have them in multiple places because less work to move it from the root meristem into the roots, and from the apical meristem all the way down to get the same effect. Sometimes things are moving that far because quantity needed needs to come from other places. But by dispersing it throughout the meristematic tissue, just like the plant response of I can make a root or a shoot at all these locations, now I can also manipulate my own growth response at all these locations because the hormone is easily accessible and proximal to make the effect happen. Hormones rarely act alone. The responses are achieved by a combination of interactions or crosstalk with hormones and other signals. So we talk about it in kind of a mechanistic way, like auxin does this. And to a large extent that's true, but just keep in mind that there's always context. There's definitely a relationship among the hormones. It's about the relative quantity of each one, not just presence or absence in most cases, and their signals are working in tandem with the plant observing, sensing what the outside conditions are, and thus what its needs are to regulate the amount of hormones that are going to be released and the type to get a specific effect. So now I'll introduce them and we'll get into what those effects are. These are the classic 5 plant hormones. Like I said, there's others out there with more minor roles, but these are ones that exist in higher quantities, have clear effects, and ones that we commonly recreate in plant management synthetically to use in greenhouse, nursery, and agricultural production to get a specific result. And those are auxin, cytokinin, gibberellin, ethylene, and abscisic acid. So, you may have heard about these or a couple of these before, and now we're just going to take a look at, for each one, what it does, where it shows up, and what its relationship is to the others. These are the big 5, and I've divided it into 2 groups, the growth promoters and the growth inhibitors. We've got 3 growth promoters, numbers 1 through 3, auxin, cytokinin, and gibberellin, and 2 growth inhibitors, ethylene and abscisic acid. Growth promoters first, and auxin. This is maybe the most well-known hormone, right? It shows up in a lot of plant management functions that we're doing both like here, you might be doing at home, etc. And one of its primary roles in the plant is controlling apical dominance. I introduced that idea of apical dominance when we talked about stems several weeks ago. And so a refresher here is how auxin, think back to just a couple slides ago, we said this appears in meristems, right, including the apical meristem. So here's our apical bud. And this apical bud, these leaves haven't happened yet. Down here, some old leaves, these buds at this node, and these buds at this node, are being controlled by the auxin quantity from the apical meristem. Some of that is coming right down to these buds, and the further you get, so let me maybe space these out a bit, the further you get from the meristem, the less effect it has. So this set of nodes is being significantly inhibited from shoot growth by the auxin. And this one is being slightly less inhibited, so there might be some bud break here in a little twig, and this one less, to the point that we saw actual leaf development. And then as you go, right, maybe this one's even bigger because there's even lower quantity of auxin reaching it. So auxin has this role in apical dominance that inhibits shoots. So that means stops those shoots from growing. Right? sometimes we want to maintain this feature because we want an open top. You like the big look of the leaf at the bottom. You are concentrating on maybe this stalk getting taller. We want more vertical growth. However, oftentimes we want the opposite, especially in greenhouse production or in a pruning situation. We might want a bushier looking plant with more leaves, more vegetative growth that looks fuller, that people are going to think they're getting a better value for, and is robust, like large in size, but not tall or leggy. And so that's then where we break the apical dominance, similar to like breaking dormancy, and you prune or pinch out that apical bud, and then the resulting effect is going to be now that auxin producer is removed, and we're going to get that result leaf, or we're talking about shoot production, so let's make it a shoot. Eventually make it a leaf out there that actually develops, and then likewise this one's not inhibited now too until this spot, right where we pinched, right above the node. This is now the new site of apical dominance, and auxin produced at this node is now inhibiting this shoot, right? And a little bit inhibiting that shoot. So that whole process repeats just further down the stem, and that might be on a plant where you're pinching several shoots, right? That same thing is happening every time. So by pinching, you're making it push out. By not, you're letting it elongate. And so that can dictate, right, or you can dictate with that knowledge the kind of response that you want. Maybe you want more airflow, and so bushy is not good, right? You don't want to prune. Or you want to prune early to get a bushy flush, right? And then you want to let it go. Or the opposite. So it's a simple way to impose your will on the plant's development at each branch, at each shoot, because of this inhibitory effect, which we call apical dominance. The top one has the power, has the control. So that's a major role for auxin. So generally, it promotes increase in cell elongation in shoots. Yet, I would guess most of you, if you heard about auxin before, you heard about it as the rooting hormone. You heard about dipping your cuttings in auxin to help promote rooting, and this is likewise the other major role for this plant hormone auxin is root development. So this is, again, helps this at the cost of shoot development. So that's like, you're taking those cuttings, you've cut them at the node, and now it has no roots, so you're using plant hormone to stimulate root production more quickly by having a higher concentration. Many of you used synthetic auxin in class here or elsewhere? Mm-hmm. And you know that it comes in different numbers usually, right? It's like you can get the 8 or the 4, and that says something about the concentration. The higher number means more concentration, means likely faster rooting. So really important role.
That kind of depends though, right?
What's that?
That kind of depends.
Well, definitely depends.
You can be burned. Like if you put root hormone that's too strong, it doesn't have like that effect.
Well, generally, the worst effect there is just you're spending money that you don't need. But the plant's either going to be signaled to produce roots or not. So sometimes the higher concentration is required because something is less likely to callus, to form this kind of gooey chunk at the bottom and then put out little roots. And sometimes you don't even need synthetic hormone, things will just readily root on their own. But generally, I would say you're not going to compromise your chances by using too much. You're just gonna spend money. Yeah, Joshua.
Can you help me understand if it is inhibiting the elongation in the roots?
Yeah.
How is that helpful for it to be used as a rooting hormone? Less growing long means more dividing of cells?
Not quite. It'll make more sense. I need to explain cytokinin also to get the reverse order, but this is in existing roots rather than in a cutting, creating new roots situation. So this is like, you'll see, it's the reverse role of cytokinin, which encourages that plant's roots to grow. And that's why auxin is generally produced at the apical meristems, right? Because most of the time, we don't want to inhibit root growth, right? In the existing growing plant. And it does seem like a weird situation, right? That it both does that for existing roots and yet encourages new roots to form. But that's what it does. And it's important to note that here, in these cuttings, it encourages both the callousing, that's the production of meristematic tissue at the wound, at the cut site, and then the initiation of the root. Andrew, go ahead.
Just a practical question. Whenever I purchase a rooting hormone, it doesn't say auxin in the ingredients list. Is it called something else? Or
Yeah. Anyone know what it says? Rooting hormone. What's that?
I said Ormex, that's like.
Hormex is like the brand name.
That's what I thought.
But you'll see something like IAA oftentimes. And that is just the synthetic version of auxin. Julian, and then
I've never used anything like this. Is it just like introduced through water? Is that how you get the hormones into the plant?
if you're using this rooting hormone like in a cutting situation, yeah, it's a powder. And so yeah, you dip the stem in a little bit of water, make it moist so that the powder sticks, and then you dip it in the powder and then put it into whatever media pot cell that it's going into. Yeah,
So
Kayla?
we use the auxin when we were in the propagation class, and we pretty much only used it for like woody stems. They're more common just because like the softer tissue has an easier time like producing the roots.
Yes.
Certain
General,
situations?
yeah.
Okay.
And in general sense, yes. And there's species differences in how readily they root, but also there's time differences. So that woody plant, when it's dormant, it needs more time and maybe help from additional rooting signal from auxin to get going.
To like kickstart it.
Because it's not actively growing. In comparison, you can see this is like a green shoot of begonia or something, and it's already in active growth, so it's more, you're going to get a more immediate response.
So the auxin are to be present there then? I mean, I guess auxin would be present in both, just like one is not really utilizing it.
Yeah, it depends on what the rest of your cutting looks like. Like it might be only another node with a couple leaves, and then there won't be much.
Okay.
So it's a
Just
little
my
bit
observation.
dependent, and then the conditions that the cutting is in also matter. So warmer soil temp is going to make that development faster, colder slower, and likewise the ambient temperature has the same effect, and the humidity of the air around. If it's humid, that's good because this little thing doesn't have any roots yet, and if it's dry, then that's going to further stress it. And so there's going to be energy expended trying to deal with no water at the expense of making roots. For sure.
Is it the idea for the hormone to have physical contact with the meristematic tissue itself to cause that, or can it be absorbed into the plant
at the wound, when you dip it, then it can start moving. And so at that point, if you've cut, here, there was no node, or you're losing meristematic function because you just made the cut, so that meristematic tissue reaction is going to be less. So you want to contact that if you can, but also get it in.
One more question.
Mm-hmm.
So the liquid one is the same thing?
Yeah.
Okay.
Yep, liquid or powder. So the liquid ones are basically the powder just dissolves. And then you just one step, just dip and stick without the secondary into the powder. Those are pretty nice because they're taken up more readily than the powder because the powder still has to dissolve. And it's easier to not use too much because it's just kind of what sticks to the stem. Instead of with the powder, it's easy to kind of lump it on there pretty heavy.
It's way more expensive.
The liquid is more expensive. Yeah. Yeah.
They sell it like an ounce or two.
Yeah. You're paying for convenience there and for likely quicker uptake and thus quicker response. So, big two here with auxin, root development, shoot inhibition. It also has a role in phototropism, which is the plants follow the light effect, and I'm going to talk about how that works too. And at really high levels, it's used as a herbicide. You might have heard of 2,4-D. It is a synthetic auxin that's used to kill broadleaf plants. While I told Dee that there wasn't danger in the cutting situation, right? It's because there's no roots and we're just trying to develop them. And it's also the concentration is much higher, right? In Hormex number 8, say, which is like a concentrated one, is 0.08%, right? So less than 1, right? Many times less than 1. And the herbicide levels are like 100 times more than that. But it does have that effect where it both inhibits the roots of that plant, that developed plant, to Joshua's question, and the shoots, right? So it's stunting growth at really high concentration.
But we said that auxin promotes root development.
That's, I know.
Here it is with the excessive concentration.
Well,
With existing roots, yeah, but if it doesn't have roots, then it promotes roots. But am I saying this right? If it doesn't have roots, it promotes
So in the case
roots,
of the
right?
cutting that's not yet rooted, it's promoting rooting. But
If it's
in
already
an
has
existing
roots.
plant, it's reducing the cell's ability to expand, to elongate. John?
Please help me make sense of the fact that you're saying it inhibits shoots, and the bullet point says promotes an increase in cell elongation of shoots. To me, those seem like opposites. They shouldn't exist together.
That's right.
So what is wrong with
Yeah, that would make sense of that. So inhibits shoot growth and promotes an increase in cell elongation in shoots.
Does that have to do with the apical meristem? So like if you top it, then it does promote the shoots, but if not, then it doesn't?
Yeah, it's close, yeah. Julian?
I was kind of thinking of the fact that it's not a true inhibitor, like it's not naturally inhibiting any of the root growth. That's why it helps it when there's none of the cytokine present, or at least less of it, because there's not as much there. So it's like just promoting any kind of growth, and then once the cytokine is present, it's antagonistic to that, and then that's when you get the inhibiting effect.
Yep. You're getting at it. There's a direct antagonism between cytokine concentration and auxin, and as it switches quantity higher in one, higher in the other, it's going back and forth. But part of this, John, is the opposite of the root situation where here, these buds that have not yet, they're not yet a shoot are being inhibited from becoming a shoot, but existing shoots,
So on the board you have there, if I just added in inhibits new shoots,
yeah.
that would apply.
Mhm. And Andrew,
Yeah, no. that was
Same.
just- yeah.
all right, so on to number 2 is cytokinin, and you'll notice a sort of antagonistic or opposite relationship here with what I just said about auxin. Generally stimulates cell division, and we're talking about growth promoters, so in a crude sense, they need to either promote cell division or cell elongation in order to promote growth, because those are our 2 methods of growing, is make more cells or make the cells we got bigger. So these stimulate axillary bud growth. So those new buds that you're just asking about, growth from cytokinin, stimulates lateral shoot production and delays senescence, and it has this antagonistic effect to auxin. So when there's more cytokinin, right, you're getting these signals to grow more shoot and signals to inhibit the effects of the auxin. For sure.
Do I understand that correctly, and kind of like the last slide where it's not as correct to say it stimulates axillary bud growth as much as it stimulates formation of axillary buds? Because where you said the auxin was stimulating cell elongation in shoots, or those were only existing shoots. So like, does the cytokinin more like, how's it to stimulate to form them as opposed to like?
Yeah, it's the actual development of the bud, right? Like making more cells that makes it from one little thing into a whole bud.
So like a shoot growth and a shoot formation are just very separate events that are happening.
Yep. Yep, so here, yeah, we're talking about the actual development of the bud from a tiny little, right, thing at the node, which we called, leaf primordia, apical bud, right, and then now expanding with the effect of this hormone. So cytokines have more mitosis, that's cells dividing to make more cells. And more cells means more plant growth, the growth promoter. And here is a look at the interaction between these two. They've got, so auxin on the left and cytokinin on your right. It inhibits axillary bud growth, auxin, and favors the root development, and it's generally moved downwards from the top. Cytokinin generally moved upwards from the roots and has the opposite effect. Right? It encourages that development of buds and shoots and reduces the root growth. So as the quantity changes, the effect can change. Whichever one is in more supply at a certain site at a time, that is the signal that's getting started. So more auxin, it's going to mean more root development. And then, if that changes, that can stall out, right? And then the new signal says, don't worry about that, start making some shoots. And so we can use quantities of these things to force one way or the other, right? The synthetic auxin gets us a higher concentration of that we say, hey, you little cutting with no roots, your focus needs to be on getting them so you can absorb water, and then we can worry about making shoots. So you can also then, you can add synthetic cytokinin and get more shoot growth down the road, but with the cuttings, right, vegetative cutting, your focus is on first developing roots, so all the rest, getting water in with nutrients, can follow from there. So these two, when you think about the auxin and cytokinin, you think about them together because they have this antagonism, root and shoot, promotion or inhibition, depending on the quantity. And our third growth promoter is gibberellin. And this one, so with each thing, as I've said, you try to distill it down to what matters here. Hormones affect plant growth. Auxin, roots, promotes roots. Cytokinin, shoots, promotes shoots. Gibberellin, just growth, just promotes growth generally. So think gibberellin growth. Right? General growth and cell elongation, especially in stems. You can get some really elongated internodes with this, but not exclusively there. Right? It's part of the germination signaling of seedlings, dormancy breaking after the imbibition, that taking on of water. Then gibberellin signals inside the seed are one that codes for, hey, situation looks good, sufficient water. Right? Let's start the chemical ball rolling of development and getting that radical poked out into this moist environment. You can change flowering time a little bit with it. It stimulates fruit production. This guy, he's showing you comparison of grape yields treated with gibberellin at different concentrations and not. I mean, and obviously, right, the treated one has more fruits and larger fruits. Steroids, yeah, plant. Right? It's like when we take human growth hormone, right? We develop more. This is plant growth hormone. This one too, Andrew, like you were saying before, you'll see it called GA on the bottle, on the label.
That's more straightforward.
Yep.
Is that considered organic if you're applying gibberellin?
It depends. There are some products with all of these. To that question, you didn't hear in the back. Your question was, is this organic? And the answer for any of these is it can be. You want to look for the OMRI approved version of the thing, which is like the agency that certifies products for use in organic rather than like farms themselves. They're based in Eugene. And you'll definitely notice a price increase with the organic ones, but they're out there. However, I'd say most of the time it's majority synthetically derived. It's cheaper to make, and you get more effect. So gibberellin growth, auxin roots, cytokinin shoots. Christopher?
Did that size difference in the grapes happen because they introduced gibberellin earlier than the plant normally would have, and so the fruit had more time to grow large, or did it just happen at the same timeline and it just caused more rapid cell production of the grapefruit?
Good question. In these studies, they were applying it as the once the fruit was initiated. So once the flowers were open, and then pollination occurred, and then just adding synthetic gibberellin. And I don't know the concentration that was used.
So it literally makes it grow much faster. It doesn't just cause the plant to start doing it when you want it to earlier in the season.
It can do both of those. Yeah, the second one is the altering flower time. So higher gibberellin pre-flower can speed it up. Yeah, Zach?
With this accelerated growth, are you gonna run into like issues with like say with like farming and like affect the taste or affect like nutrients?
I don't, taste-wise, I don't think so, because it's all the same cells, just getting more of them faster and longer, bigger cells. But primarily, even conventional farms are not like just spraying gibberellin out there to make every grape 3 times the size. Part of it's a cost issue.
It's expensive.
What's that?
It's gonna be pretty expensive.
It'd be pretty expensive, and then you guys still gotta pay somebody to do it, right? Mostly this is used in controlled greenhouse production, and I'm more talking about it as the actual hormone that promotes growth that's produced by the plant itself. So all of these are plant-produced hormones, internal production, and we've figured out ways to synthesize, like a synthetic approximation of them that then we can use to say, like in greenhouse production, for example, with flowering time, you might want to, treat one same crop and get early flowering, then that can spread out how long you're selling X, Y, or Z product with flowers available, you know, without having to do 2 plantings or 2 seedings or whatever. So there's some uses like that, but mostly this is just, here's the growth hormones in plants and their effect. Yes?
Does that cause that plant behavior, despite the balance between the other 2 hormones?
Yes, there's, good question. So while there's a direct antagonism between auxin and cytokinin, gibberellin's working separately. So it's its own production directed at various sites and not directly influencing either of these effects.
And vice versa, those are not affecting you.
Likewise, yep. So it's targeted in auxin and cytokinin to particular organ system, roots or shoots, and in gibberellin, it's generalized. And so wherever, you know, needed in certain quantity or applied by people, then that's where you get the effect. Inhibitors. So let's take a break now. Also, at 2 o'clock.
I don't know how I'm doing this on the class today. We got one more. It's like, wait, another Oh, that's right.
Always curious, what you're gonna ask. There's so many ideas that come up, like this,
Zach, you're going next week.
Oh,
Yeah.
friends do. Good. Apocalypse style, so cool. I know, it's very good. Huh?
Like west of here, or like?
Yeah, so we're on the East Coast, so great. We did that. Just like, figure out all the this area.
To get to it?
Well, I stopped.
There by honey. This has full stuff.
Appreciate it, but I don't want to take away
Oh, absolutely. I read plenty, yeah. Because I think would make a couple different sizes, just like kind of smaller. Plus, you can bring all sorts of stuff.
Soft, good flavor, nice and wet.
This is almost, I like the small size about it, also. It's the bacon piece.
If I throw a bunch of gibberellin on the strawberries, it helps me.
I would love that. It's not, you know, it's not funny, but obviously.
Also, I didn't hear what they trying to get some wild they call them like mountain, but wild coast. That sounds wild
And I was like, I know. Every time I'm like, I know.
Alright. Continue with the growth inhibitors. Keeping track of your promoters, auction setting, managing gibberellin. Maybe they're either making more cells, or making cells bigger, or they're both. And inhibitors, ethylene and abscisic acid, have different effects. So, we're calling these growth inhibitors. Ethylene is a widely used one for promoting fruit ripening. We talked about with gibberellin, moving the flowering time earlier, that application. And here we're talking about ripening the fruit more quickly. We mentioned when we were talking about parthenocarpy a couple weeks ago with seeds and fruits about bananas, and they're an excellent example of synthetic use of ethylene. So bananas are all picked green, and then they're put in plastic bags with ethylene gas, and then when they sit on the ship or the plane, they're slowly ripening in transit as they're getting to the, like intermodal hub off the ship, onto the truck. The whole time, they're just sitting in ethylene, and then they're like, hopefully light yellow. When they get to the grocery store, that's their goal. And then even more yellow when they get to you. And so, you might say, this literally says promotes fruit ripening. And the way that's reconciled as an inhibitor is ethylene is pushing this plant toward death, for the process of reproduction and successful fruit ripening. That's going to mean that seed can now be dispersed. And right, in a lot of plants, especially annuals, but across the board, like in bananas, for example, they're not annuals, but you need a new shoot for next year. That leads to senescence, and that is the actual degradation process, the breaking down, and the eventual dying of the plant. So, fruit ripening, on the one hand, very useful. And there's lots of synthetic ethylene products out there that are getting use, not just in commercial agriculture, but definitely in greenhouse and nursery production, used to abort flowers in high enough concentration you can get rid of them. That's how commercial apple orchards do fruit thinning. In the springtime, apple trees produce lots of flowers, and they're thin. The flowers are removed to the tune of 80 to 90% of them. So they're only allowing those trees to ripen 10 to 20% of the flowers there. And so to do that on a commercial scale without having to climb in, and I don't know if you've ever hand done that, it really takes a long time. And so they spray something like this at high concentration. It makes those flowers fall, right? It pushes them towards senescence. They're not fruit yet, so it's not ripening fruit, but it is making that leaf basically age faster, and then, or that flower, excuse me, age faster, and then drop off, and leaving a much lesser quantity of apple flowers to pollinate, and then support for harvest. So that's one, what's that?
But why?
But why?
Why do they do that to apples?
Because if you, it's an energy distribution concern. If you let the apple tree ripen every flower into fruit, you would get a bunch of hard little apples and not like a big red, delicious one. buds and promoting lateral branching. You can see here in these flowering plants, no ethylene treated with ethylene and then an ethylene inhibitor, right? It goes back to its no ethylene look, and with just ethylene and all that flower drop. So sometimes that's the effect that we're looking for. We want to speed it up, or right, we want to go and speed up the fruit production. And I mentioned that post-harvest fruit production role, and it also is a contributor to leaf senescence, which is that leaves falling in autumn that we've been talking about. We talked about the plant pigments like chlorophyll and carotenoids, and how the other pigments show up behind the reduced production of chlorophyll in leaves in fall. And likewise, there's ethylene signaling that makes this happen. So regular maintenance phase, often being produced in the leaf. As things get later into the fall, then ethylene starts being produced, especially down here at the petiole. That signals a sealing right here, right? A division that cuts it off from the vascular system of the rest of the tree, and eventually then the combination of wind, gravity, and external factors, along with the fact that now there's like a dead layer right here, eventually means that they fall away. Yeah.
Is that the same signal if it was to give it to like a tomato to cause it to, the fruit to start ripening by sealing off the connection to the plant?
Mm-hmm.
So it just kind of does that, that's its behavior that it does.
That's right, yeah, so you could use the ethylene for the same purpose there. That would be like ripening the banana, it has the same effect. And so, and for those, it's fruit, right? And this is a leaf. But in both cases, it's senescence, it's driving it forward in its development process faster than it would otherwise. And
On that previous slide, even though ethylene is an inhibitor, could you use it to get like double the growth in a year because it's promoting lateral branching at the expense of flowering? Or is that kind of like a one-way ticket into senescence for the season?
well, I don't know that you could necessarily get double growth, but you can definitely get a flush of faster growth out of it. It matters what your end game is. If you're selling a thing that's vegetative, doesn't matter if there's flowers on it, maybe more is better, right? If you're selling a potted flowering plant, you gotta have it. Or convince people with some buds that are starting to open that they're gonna have it soon.
Or you want to get to like a 10-gallon version of a hydrangea in one season as opposed to 2 or 3 weeks?
Right, yeah, you might be able to do that. So you were nodding, do you have experience applying this?
I'm sorry, what was that?
Do you have any experience in applying
Oh, yeah,
it?
I use Florida all the time.
Yeah, you wanna talk about maybe one example of how you use it?
I know they do it on, what was it? I think it was some of our fuchsias to like have them drop the flowers early on or promote branching. We don't do it on all of our varieties though, because like I think it's like I forget which variety we have, but it does not interact well with it for whatever reason. But yeah, then they'll grow out, branch, and then flush out again by the time it so. Yeah.
It's delicate, right? 'Cause
Oh, yeah,
if
no,
you get it, too much, then now it's all branches.
Exactly. Yeah. But that
So-
last season.
Yeah, fuchsias is a good example, because early on, the focus is going to be pinching, right, to remove that auxin and get more branching, so that later, there's more opportunity for flowers. So by initially knocking out the existing flowers, right, you're getting a better, bushier, more flowering result later. So,
Oh, we also use ethylene to defoliate our hydrangeas too, when we put them in the dormancy earlier. So by the time we want to sell them, they're flowering before.
here you go, yeah, a time demand indicated that it would be helpful to have this thing sooner, so push that process forward, right? Drop the finished flowering, right? And now you can move it into cold storage, or we can sell it, or whatever.
Yeah, we move it in the coolers to get the colder weather that we want
Mhm.
originally.
Yep.
That's early.
that the only way?
No, you can also do it, so the way we defoliate our, hydrangeas is we put them like in a big, like truck container or whatever, and we do it through like diffusion of like gas.
Kind of like how they do bananas.
Yeah,
We went to
yeah.
the organic farming
Yep.
place
Yep.
and they got these big things that put bananas in, pump it with ethylene.
Yeah.
That's what we do with the hydrangeas.
A few of you, anyone else was at that food harvest field trip? Yeah, two things going on there of note connected to this. One was the whole banana ripening thing, right? It's like these big garage doors, you slide in your pallet of bananas, and they release ethylene gas. Most of the time it's applied as a gas in this closed room, and they try to get them from that like green to the 3 on that. You saw like the chart there, 1 through 5. 5 is like, you know, yellow to browning, right? That the grocery store wants by applying the ethylene and then managing like, okay, it's been there 24 hours, generally that gets it from 2 to 3, and now we ship out. And then the other thing was, so this is organically grown company, they're like a organic wholesaler, so the middleman between the farms and commercial grocery stores. And in the rest of their warehouse, where they're storing potatoes, they're storing onions, they're storing carrots, all these other things, they have ethylene scrubbers.
Yeah, they don't want no
Right?
ethylene.
They want no ethylene there because that's going to age that produce, right, more quickly. And they have low temperature so that the respiration rate is low. Okay? And then the humidity depends on the specific crop. Yeah, good connection. Leaf falling is connected to senescence, even though abscisic acid sounds like abscission, sounds like falling off. Turns out that's not the one immediately related to leaf falling. What it is related to is inhibiting seed germination. So if you're looking to stimulate some seeds and inhibit others, you can add gibberella to get it started. You can add abscisic acid to cut it off. It suppresses bud growth, and it mediates plant response to stress, especially drought, and it signals closing stomatal guard cells. And I mentioned no specific role in abscission, but here, right, we talked about the importance of filling that vacuole with water, making it turgid so that stomata can open, gas exchange in and out. That signaling comes from abscisic acid. And so that's a big role for this one because, again, millions upon millions of stomata receiving those signals to open or not open.
When would you use this?
What's that?
When would you use this?
Well,
When would it be to your advantage?
generally here, there's not, at least with the affecting stomata, there's not as clear of a commercial synthetic use. It's actually just the plant-produced abscisic acid doing that signaling. Here for seed germination, you can apply it in a liquid form. If you want to stop these things from germinating, you want to slow it down, you want to make sure they get through to the next season or whatever, you can do it that way. Yeah, Dale.
So could this have been applied to different plants when we had that super hot weather?
Yes, you want to say more about what you're thinking about that?
Well, we had that 117-degree weather.
Sure.
We lost a bunch of blueberries.
Uh-huh.
I applied this to stop the respiration of the plant.
Respiration's just according to temperature here. Yeah, this won't do that.
It's going down.
Yeah.
So the hormone will cause the guard cells to shut the stomata.
It signals to open. When it gets the signal from abscisic acid, that's then the signal to fill and
So the lack of it is the signal to close, because it says signals closing stomatal guard cells. So if it's not present at a certain level, then it'll be open.
Yep. So it's a quantity. It's like at a certain concentration it means open, and at a higher concentration it means closed.
Is
So
that
there's like,
talking to the nucleus of the guard cell, or
where is it produced?
no? Like how is it interacting with the guard cell?
Oh, like Yeah, it's an interesting, question and then a little bit complicated. But yeah, basically, the hormone signals are moving through phloem and then getting to the guard cell, going inside the cell to, excuse me, to the nucleus, where that response then is coded to like, it receives it, it's like, oh yeah, thing says open up, and then now that reaction continues. Acetic acid and ethylene, sorry, go ahead.
So this would, if I was to spray this on a plant, would it like directly just stop transpiration? Or how would that affect it?
Good question. I'm not really sure. I think that, because the plant's already producing this, it's going to try to override like our use of it to get what it needs.
Yeah.
But I think at a high enough concentration, you could shut it down for a while. And it depends too on the conditions when you apply, because when when you apply something externally, if you do it to the soil, it's going to take some time to be taken up and get used systemically. But the other way to get it in is through stomata. So if they're open, then it's going to, you're going to have some effect there. And if they're closed, not as much.
I was just thinking, so like in a scenario where it is really hot, we're getting water loss, things are transpiring, I wanted to slow water loss down. If I sprayed with acetic acid, would it slow water loss?
I mean, if you're able to make it close its stomata, then yeah,
Okay.
right?
It's probably optimized for doing it itself.
Yes, I think generally you don't have to do that because the plant already wants to do that as much as possible, unless.
I'm not saying I want to do that, I was just curious.
There might be that next little level of like, hey, you could get, I don't know, like 5% more growth by reducing the water loss on top of what the plant would do itself. That's possible.
Okay.
But I think most of the time, Andrew's right, that it's already doing it. And people were primarily manipulating the environmental conditions rather than that response.
Oh, okay, yeah, that was curious.
Yeah.
So when does the acid usually show up? Is it like during like winter time, or is it,
When does acetic acid show up?
yeah, like?
Oh, it's getting generated, right? Definitely more in
So like
fall
winter?
into winter,
Okay.
right, for some things. But because any growing plant needs stomata open and closing all the time, there's always these hormones being produced. That's like one of the, and I say, respiration is like the base level metabolic functions of the plant. Producing hormones and moving them to the spot where they're needed to do something is one of those functions. So that's one of the things that the energy from respiration is feeding all the time. And if you run out of glucose to break apart and get energy, then you can't move, say, the auxin to get more roots. And then that has another set of problems attached to it.
So like, yeah, so it says especially drought, so like if it isn't getting enough water, this will simulate it to close the stomata.
Okay.
I just wanted to double check that, acetic and ethylene don't interact with each other like oxygen and stuff.
Not in the same antagonistic way. No, there's some instances where like together, because they're both inhibiting, they can make a thing happen more quickly, push it toward its senescence toward the end faster. But because ethylene is generally a gas, they're not often working in the same place at the same time.
They don't affect each other as much.
Yeah. Yeah.
So, looking at inhibiting seed germination, is this the hormone that's responsible for dormancy?
Mhm.
With the seed and the plant, like when it goes into dormancy, this is like causing all that to
Yeah.
happen.
Yeah, so this is then the one that signals like shut it down, make the hard seed coat, right? Stop, slow down respiration, don't try to make root grow more now.
And that hormone's like identical to the plants being used in the plant, like there's no chemical difference, it's the same hormone, just filled in different.
Same hormone, yeah, just different spot, and different signal set of how it's received and then like which next reaction it kicks down the chain. Yeah.
So, more of this hormone is produced before it gets signal dormancy?
More abscisic acid is produced the older it gets.
Or, the colder it gets?
The colder it gets.
Because I know like cold has correlation with dormancy because of the respiration.
Okay, so this is specific to seed dormancy now.
Okay.
Yeah, so not across the board for plants at temperature. So those are our big five: three promoters, two inhibitors. Know, the primary role for each one. So like roots, shoots, general growth. Ethylene, it's got a couple things, but we can say senescence, abscisic acid, stomatal control. Here's some other examples. You might hear about some of these things. Fluorogen's always one. It's kind of talked about in like ethereal terms. Like it's a mystery out there that there's something that is suspected. There's one hormone that controls flowering signals to yes or no, flower or not. But it hasn't been worked out quite so much before. So just examples. There's others out there. I'm not going to ask you about them or tell me specifics on any of that. Here is kind of a summary chart. So as you study, this format works for you. It's an option. I've got them laid out, where they come from, how they move, and what they do. All the stuff that we just talked about, but put it into a digestible piece for the people who either charts or words are the thing. Here's another way to look at it for more visual. So hormones here, and then green check mark is like promotes germination for gibberellin, promotes growth, promotes flowering, and it doesn't have a role in abscission or dormancy. Obviously, it would be the opposite of germination to promote dormancy. So another way to look at what these things do. You can see like there's a transition from promotion, inhibition, and then ethylene sort of rides the line because it's developing those fruits, but it's also pushing senescence forward more quickly. So just trying to condense that info and make it easily digestible and boil it down to like when you hear about these hormones in the future, you get the function, like in a couple words, the function, so you can just talk about that or know like, hey, I'm trying to root these cuttings, I need auxin. Questions there? So we're still talking about growth responses that are hormone regulated. The rest are some combination of the hormones that I've just talked about. And we call these growth responses to environmental factors tropisms. A positive tropism is toward a stimulus, so the plant grows toward the light, that's phototropism. And a negative stimulus is away, or a negative tropism is away from a stimulus. These are hormone-regulated responses. Phototropism, I hinted at earlier when we were talking about auxin, that plants grow toward the light. Sun hits the plant at an angle, auxin moves, it causes cell elongation on the dark side. Basically, auxin runs away from the light, gathers on the dark side of the meristematic, apical meristem tip, and that all that extra elongation makes the thing tip over from the back and look like it's now curved toward the light. So stems are positively phototropic, they go toward the light. This next one. So the pink is the auxin molecules. Direct overhead, there's no change because it's straight on. Now at an angle, auxin's migrating away from the light source to this side where those cells are elongating due to the effect of the growth promoter, and that's making it those long cells are tipping the whole thing at the light source. So it's an effect of auxins escaping the light and then growing on the other side that makes it head toward the light source. And of course, that's happening throughout the day. It's changing.
So elongation is reversible, is the implication.
Well, now it's going to be like that, and later, right, if the sun moves, I mean, elongation, you can't, once the cell elongates, then the cell wall can't be recontracted. So that cell is now longer, bigger, more volume and area than it had before. But it can then, right, if the light source moves to the other side, then the thing switches. And so maybe there's more growth at that time, which makes it tip, or maybe it can't quite tip all the way because that side's already longer and then it looks not so much. But we tend to, in the course of a day, right, it's mostly moving, as the sun goes generally east to west. It's not like 360ing around, right? It's just like a gradual movement from one to the other. Guys, feel the sunflowers, they're following that light. Lots of oxygen moving around there. So that's phototropism, following the light. Gravitropism or geotropism is following the gravity, following the earth. Shoots are negatively gravitropic. They go away from the stimulus, which is the pulling down of gravity. They grow up, and roots grow with gravity down. You already knew that, but now there's a name for it that the plant can actually sense how it's oriented, and if you flip it over, it's going to still try to do this. And that looks like this. So here I'd say this plant has recently been upside down. When it was the other way, it was trying to grow right side up, but now that looks wrong. Right? And over here, it was just put 90 degrees on the side, and quickly, you can see, it figured out, oh, now I'm horizontal, you gotta start going against gravity here. So it can sense gravity, and then it moves in relationship to it to make sure shoots going up, because plants understand that that's what the light source is, and roots then corollary go down. So what you can't see in here is likewise the roots are starting to move this way as well. Lee?
So I was kind of going to ask when you answered, but in the one that was upside down, this is just because it's funny to me, would the roots eventually start growing out of the container it's in if it stayed upside down like that? Like if the whole plant didn't eventually fall out, would it eventually they start crawling out?
I mean, if it didn't just fall down.
Yeah.
Let's take a look. So let's say, so that pot is upside down like this, right? Now this tomato it started like this, and then it figured out, oh, I'm upside down, so I'm gonna start going like this, right? Meanwhile, for roots, we're going like this, and they figured out I'm upside down, so I'm gonna start going like this. I don't think it would be like they're jumping out at you, but yeah, they could possibly come this way, but they probably air prune when they hit the surface. It's, they're not just gonna hurt it down. But yeah, that's the concept. Julian, was there a hand back?
Yeah, I just wanted to be clear, it's not just sun, gravity is an actual stimulus that's giving.
Gravity is the stimulus, yes. Or geotropism has that name because the Earth, it's like the pull, right? The gravity of the center of the Earth, everything is being drawn there, and that's the stimulus that's getting
So it's the effect of gravity, not necessarily the plant sensing gravity, because that kind of blows my mind if I'm to think of it in that way. It's I don't have the sense of gravity, maybe the proprioception.
Oh, I think you do.
I do, but it's.
You know when you're upside down, right?
Right, but it's hard to relate that to a plant, so.
Yeah, you're not gonna then start growing differently, but Yeah, they're sensing that pull.
So it's not just gravity happening, it's a sense of gravity.
Yep. Yeah, so,
Do we know like how they sense gravity?
it's a little more complicated. There's, I don't think it's so clear.
Okay.
You know, and I don't have, like a crystal,
Yeah, but they don't have worry.
explanation of it, right? That's the perplexing part about them, right? There's no, central nervous system, yet they make all of these complicated responses to the environment constantly, and they can do it at any point, right? Well, at any node, right? At any meristem. So yeah, there's something there that we don't know. Have you, or was it?
I was just gonna say, those upside down tomato plant bags.
Yeah. What about them?
Well, that kind of what we're sort of talking about when I talk about.
So have you tried those tomato upside down bags?
So now, the tomato's gonna come down, it's coming down, but it's gonna start growing up, and then the roots will go closer to where the tomatoes grow coming out. You see them all over the place.
Yeah, I mean, I don't know. Has anyone else grown those upside down tomato bags? Yeah, I mean, you think about this setup, right? The plant's not the only thing responding to gravity. Right? All this water is also going that way, right? And then what's getting tricky for the plant here is that it wants vascular system, xylem, right? It wants to move that water this direction toward the top, and now, as this curve happens, now it's got to push this water around and back up, and that's going to be a chore, right? That's going to be extra energy. Christopher?
So speaking on the tropisms, and you mentioned the air pruning, and that got me curious about the last slide with oxygen, and the air pruning is that the oxygen going away from the light and causing the root cells to grow in a different direction, like you were talking about the flower facing the sun in kind of the opposite sense, where the oxygen's going to the dark side and causing the roots to grow towards the dark.
Yeah, so again, whenever we're talking about something with oxygen or cytokinin, it's about the relationship. So then there it's like, yeah, we're inhibiting those roots from growing more because then it's just gonna dry out there, right? So that's then more oxygen, less cytokinin that's giving that signal. Yeah. One more, Caleb?
I mean, I was just gonna say, I was curious about the gravity. Does it have anything to do with like the oxygen as well? Like if the top of the plant is seeking oxygen, so it wouldn't like go the other way? Not necessarily.
No, because there's oxygen in the soil also,
yeah.
in the soil pores. Yeah. Marina?
So if you have a house plant that's like maybe growing more horizontally, laterally, can you tip it and force it to maybe go the right way or without using something like a moss pole?
Has anyone tried that?
What? What do you mean, like, said?
So, like a house plant, I'm thinking of like a philodendron, for
Yeah,
example.
it's just kind
that
of crawling along
has-
a table or something.
Yeah, it started to grow like this, even not like leaning on anything, just starting to grow like this versus like this without any type of support. If you were able to tip it in a manner that it would come back upright on its own. If there was a way to do that using either of these.
I think so.
Yeah.
I mean,
Mine just wants to.
depends on the size and the conditions, like how quickly that response is gonna come. But yeah, you're talking, think about plenty of people, right, have just like philodendron or whatever, it's just crawling along laterally. But each time it puts on a new leaf, and when that one comes out, then it comes up here, right? Like this is each time, right? So it does still know which way is up. And likewise, if you then flip that thing and you hang it, say, so now it's like this and like this, then it is going to start to do that. Eventually.
I guess my question comes from when you specifically said that stems always grow up. In that case, it's not kind of more science. Where that question came from.
Yeah, I see what you're saying. It's
Up is a range.
up as a range, and it's still not with gravity, at least.
Yeah, because isn't it like weight? Why is it the weight that makes it grow sideways? In the plant? It's like it started growing up. When it first started growing, this plant was growing straight up, and then it slowly started going sideways.
Yeah, I mean, part of it is weight, right? It's still responding to gravity. And part of it is just like the genetic growth habit of those plants. Like they're viners. And so they don't go up in that same way. They have like a prostrate habit and then it vines out. So here's another tropism. Thigmotropism is contact or touch. Tendrils on vine plants are a great example. So as it touches the stake, then the response is to go toward that stimulus, toward the stake. So then wrap around it, grab onto it, and curl, and that gives it support. So back to, we were just talking about vines. Many vines have this type of response that they go toward the stimulus of something present that offers support. And so that's how then you can make use of a trellis, right, or a stake or anything, is by taking advantage of that. And of course, roots grow away from a touch stimulus. Like if they encounter a rock, they respond by going around rather than toward, which is what the shoot of that same plant would do. So that's
Why wouldn't it grow up a rock?
why wouldn't a root grow up a rock?
Oh wait, rock, oh, root. I'm thinking, pretty much. I was thinking.
It's, rocks don't hold any water.
I was thinking stuff.
Yeah. Some rocks can. And there, I mean, in like a porous rock like a pumice or something, there'll be plant roots in there. All right. Control of flowering is a really important signal. And I mentioned that we don't quite know exactly the hormone relationship, but we've got a good idea about how the plant senses daylight and then creates or negates the signal to begin flowering. So plants, I will ask you this, sense the dark period. We call it photoperiodism. We say long day plants and short day plants, but plants are sensing how long darkness is. And when we say long day plant, that means it flowers when night is short, right? Shorter than the critical photo period. Often it's about 12 hours. I'm not saying it's always exactly 12 hours, right? But most of them are in the 11 to 13 range. So long day flowers on short night. So the trick here is keeping in mind that it's sensing the dark period, or that long day means also short night. Right? The long day and short day terms that we have are, they're a little bit confusing. So a long day plant flowers on a short night, and a short day plant, right, flowers on a long night. We live about 45 degrees north latitude, so we've got a big swing between 15 and a half hours of light summer solstice and 7 and a half hours of light at winter solstice. And so our outdoor, right, forest natural environments, we've got a lot of long day plants that are flowering in spring when nights are short, post equinox, and not as many of these. And that's where the greenhouse industry comes in to offer us some really nice options if you want flowering things throughout the year, to find the short day plants that are flashy, nice, bring like a color and light to the cloudy gray here. So they sense the dark period, and some respond to flower when it's long night, some when it's short night, and some plants are day neutral, and they flower based on total degree day development or some other environmental factor that's not just the quantity of light as it changes. What's that?
It's like an auto flower.
Like a auto flower, exactly. So that one is an example of day neutral. It just develops to a certain point, right? Enough degree days, enough heat, and then heat and light, and then it just starts putting it out, right? So, how does it sense that night period is what I'm gonna explain now. So again, photo period, by having night length, which, to be totally clear here, right? Photo period is 24 hours minus dark, or minus dark hours equals light hours. Huh? Yeah, no surprise, right? 12 hours of light, 12 hours of dark, but just, right, it's a diurnal there, so that over here, diurnal dark period, right? That means the change within a single day. So, plants do this through a sensing mechanism that is about light absorption. So they're getting signals from the quality of light, and when they're not getting those signals, then they understand that it's dark. And so that informs that's how they know how long the dark period is. So we talked about in photosynthesis, so keep these numbers in mind for just for a moment. The red light at 660 nanometers, and far red light at 730. And when I showed you the visible light spectrum, so here's 660 red light, or 730 far red light. So this is what's being sensed. And remember, we were talking about the different qualities of light. The red light, not as high energy as blue violet, tends to appear sunset and sunrise. And so there's an easy connection there for the plant with sensing time in figuring out how much is there of these things that mean it's either light out or it's not. So it absorbs this PR, for R for red, red light, and it's converted quickly to PFR during the day. So when the sun's out, this signal is always being converted to the PFR. And there's a picture on the next slide. And the other thing, PFR absorbs far red light, which is converted slowly at night into the other form. So a low PFR concentration means a short day, right? Because it wasn't a lot of time to make this in the light. It means a long night. And a high PFR concentration indicates a long day. It was a lot of sunshine to make a lot of this. And that's how the plant does it. So every time it knows it gets, okay, high PFR concentration, that's another short night. And as the short nights add up, right, the short night plants, aka long day plants, are getting the signal that it's time to flower. So it takes a buildup of signals over time. Here's the basic idea, right? So red light is coming in during the day, it's being absorbed, and at night, that PFR is being slowly converted back to PR. During the day, that PR is going quickly to PFR. So the more of this, basically, the more light there is. The more PFR sensor, the more light in that day, and thus, right, the longer the day, and the shorter the night. So it's collecting that information day after day, short night, short night, short night, short night, okay, longer, and depending on if it's a short day or a long day plant, it gets that flowering response, or it inhibits flowering response. Yeah.
In a controlled growing setting, like where you're using grow lights, there's typically an option to change it from like having a sunrise and sunset versus just on and off. Is there a benefit between using one or the other? Or a disadvantage, I guess, either one?
Well, in terms of sensing, so in either case, as soon as the light source stops, then right, there's no more of this coming in, and you're getting this PFR slowly back. So it doesn't change that part. Like if you stop both of them at the same time, the signaling would be the same. But in terms of the quality of light for growth, the giving the plant this kind of, you're easing it out and easing it in, it does help development because it's recreating like the natural cycle where it's signaled to kind of wake up and get going, and then has a chance to, it's not like a hard transition from, it could be a very efficient growth under the growth light, and then it's off, right? And now it's got to scramble to figure out, oh, I should have saved all that to do some photosynthesis later, right? So it does help growth by giving it the signal, because then it has time to adjust. But in terms of sensing daylight, it doesn't change it.
Chris, what's keeping us from just going in a cycle?
It is going in a cycle. So every day, right, red light is coming in, it's changing this thing to this, and then at night, it's slowly going back. And so then during the day, it's changing this again to PFR, and it's slowly reverting. So that cycle's going on and on and on. And what the sensing is the quantity of that over the course of the night period tells it if it's long night or short night.
So pretty much clock.
Yeah, like a clock, or more like a timer. Yeah, exactly.
Sand clock. Hourglass.
Hourglass. There we go. And a really good example of these features is poinsettia, which is a huge greenhouse crop. Some of my coworkers went to Smith Gardens 2 weeks ago and told them that they produced 600,000 poinsettias for Christmas. And we know already that these are not bracts, right? They are leaves, right? Right, are a big deal.
They're not flowers.
They're not flowers, exactly, right? They're not petals, they're leaves. And this is what we're concentrating on when it comes to poinsettia production, is that flowering. So what do you think? Do people want a flowered poinsettia? Do greenhouses want it flowered or not? Not. And why not?
Because you want the grass.
What's that?
It's closer to the senescence.
Right. Because when that customer gets it home, they want it to be good for as long as possible, right? Yeah. So, there's a big interest in this Christmas industry of delaying poinsettia flowering when we're in this environment. So, anyone know, is poinsettia a long day or a short day plant? A long night or a short day?
Just shorter.
Short day.
Short day.
All right. Short day.
It's a winter plant, so.
Short day, aka long night.
wanna keep the lights on.
So you wanna keep the lights on, right? Because we want this thing to be available for sale now or, you know, in a couple weeks and look like this and not look all open and right now the petals have fallen and it's not as pretty and people think it won't last as long. So we want to keep this long night scenario as long as possible. And so, how do we do that? Supplemental lighting, right? After, you know, autumnal equinox is like 9/22. So to get to today, that's almost 2 months, right? And so, that's the answer, is more light to make that night shorter so that it doesn't flower.
They're kind of the same way with like cannabis. It's kind of the same way with cannabis. If you don't want it to flower, keep it above 12 hours of light. It's definitely going to
That's right.
Yeah.
And so when it goes below 12 hours of light, so that's, you know, long night, right? Then the same thing. So yeah, it's a short day plan. All right. So there's more to say on that, but I'm out of time for today. I will continue with that on Monday, and we'll also have the quiz. Yeah, great questions, concerns. Again, the homework is up here, but that's not due until a week from Monday.